Thanks for joining me today for this interview, Stephanie. I am absolutely fascinated with what you're doing. My background is also neuro-related. I'm a neuroradiologist, and my job is day in, day out. I see the receiving end of diseases, and a lot of these are devastating, like stroke, neurodegenerative disease. Frankly, we don't really have a lot of solution for. So I'm fascinated with what you're doing. But I want to explore a little bit your journey along your career. You're very young for being a full professor, and your academic career is absolutely very impressive. How did you decide? Now, first of all, you want to focus on neuroscience, and then you want to print essentially a brain.
Yeah, well, I was one of those kids who always, I think I asked for a chemistry set when I was 5. I was always really into science. My high school thesis was on stem cells, and dating myself, it was cloning Dolly the sheep. And when I was in undergrad, I worked in a bioengineering lab. At MIT, at the time, you could only major in biology and chemical engineering. There wasn't bioengineering yet, so I did the double. And then my grad work, I rotated in Shelley Sakyam-Elbert's lab, where she was engineering neural tissue, and I just thought it was really cool. I guess some of the more mature tissues that people transplant, I think more regularly, are things like bone, but it's very static. Whereas with the brain, you have tons of different types of cells, lots of different signaling, both electrical properties and mechanical properties. There's just like a lot to explore. And then during my PhD was also when I was actually at the Toronto ISSCR meeting where Yamanaka gave the talk describing IPSCs. And I just remember thinking, this should be a really, really big deal. And it was a really, really big deal.
So tell us a little bit about IPSC, because I don't think everyone knows what that is and why is it so amazing.
Yeah, so IPSCs are induced pluripotent stem cells. And so, again, dating myself a bit, when I was in graduate school, the use of embryonic stem cells was controversial for a number of reasons. But the reason people were very excited about embryonic stem cells are these cells can become any cell type found in the body. So in theory, you could replace any tissue. They're sourced from human embryos, which is why there was a bit of a controversy around them. And so then with the invention of these IPSCs, instead of deriving these cells that can become any cell type from an embryo, they actually can do it from a skin cell. And then now, with the project we're working on, they actually do blood draws from the patients that my collaborator, Dr. Nygaard, sees in the clinic. You can do a blood draw, you treat these cells with certain factors, and they go back to essentially your blood cell becomes a cell that can actually form tissues like you find in the brain or spinal cord or skin. And we mainly focus, for the bioprinting stuff, we've been mainly focused on neurodegenerative disorders. And the one interesting thing about the IPSCs for neurodegenerative disorders, which Dr. Nygaard's group, I think, has just published some nice papers on, is you actually, if you take these cells from your Alzheimer's patient and grow them in a dish, and you age them, they actually get more Alzheimer-y over time. You actually can track that. So
that's really amazing, because I've been thinking about that lately, about the time factor. In biology. And I don't even know if we have had any models to simulate time, to age something faster. Let's say something like neurodegenerative process takes 5, 10, 20 years. How do you simulate that in the lab? I think that could be a potential breakthrough for...
Yeah. Yes, it's really interesting because as you grow the cells, like they'll secrete more tau and amyloid beta protein, and you can measure them using assays. And so the idea was we could, first of all, grow them in 3D. Another interesting thing about some of the modeling, just because of how the field's developed, a lot of the protocols have been really focused on making like one specific type of neuron from these cells, but not modeling multiple cells like you'd have in the brain. So you'd need your neurons, your oligodendrocytes, astrocytes. And so there really hasn't been many good co-culture models. And Dr. Nygaard's group has done some lovely work where they've actually been pairing healthy with diseased iPSC-derived like neurons with astrocytes and seeing how they modulate each other in terms of measuring disease phenotype. So
yeah, that sounds like a worthwhile reading. I'd be happy to post the link of the article as well after this. But I know you guys just published a paper focusing on GBM. which, believe it or not, I did some research from the radiology side when I was in MGH, also on GBM from imaging perspective. You want to tell us a little bit about that study that you just published?
Yeah, I think, was that our Materials Today chemistry paper?
Yes, yeah.
Yeah, because we've got another one that's really exciting that I'm editing right now that we're hoping to submit. Granted, all the work was done pre-corona, but yeah, so with the cancer work, again, it's really interesting. And there's this whole idea of, the tumor microenvironment, 2D versus 3D. And what we found is when you culture the glioblastoma in 3D, they become more tumorigenic. They do form these spheroids inside of our 3D printed constructs. And the idea of that is to use that as a tool for drug screening. That's some of our unpublished work that we've been working on. But it's a lot harder to break up a tumor in 3D than it is in 2D. It's very easy to, well, not easy, but lots of things work when you're trying to kill your cancer cells when you culture them on 2D monolayers. And then when you actually have them as physical spheroids, it's much harder to break down the structure. But in GBM, as you know, it's super invasive, super proliferative cancer. So you're really dealing with these cancer masses. So essentially using 3D bioprinting gives you a way to make something more that will mimic what's going to actually happen in your body compared to other techniques.
So right now it sounds like where you are in terms of this GBM model is you've created the structure. Now it actually is a real tumor-like structure. But have you guys gone through the actual drug screening testing and then see if this actually works?
Yeah, so that's our unpublished paper, which I'm not sure how much I'm supposed to talk about. But yeah, we have we've been working with a company based out of Montreal who has some experimental therapeutics to show like what happens when you treat these 3D printed tumors with these drugs, and we can quantify tumor size. And we actually have fluorescently labeled GBMs too, so we can actually track over time. So you can just watch the fluorescence decrease as the tumor cells die. Yeah, that paper should be really exciting. And it's also really interesting because we took the model to another step. So we not only have tumor cells in the model, we also have healthy neural tissue too. So you can sort of see the effect of the tumors on the healthy tissue. And again, if you're trying to treat GBM, ideally your therapeutic would just target cancer and not healthy cells, which is why I think a lot of people know in cancer, a lot of chemo has very bad side effects because it's also targeting your healthy cells too.
Yeah, I shared your paper with one of my neurosurgical team that I work with. They're super excited about this. Currently, for patients that I deal with, they use animal models. So basically they implant the tumors onto mice, which obviously is not the most efficient process of screening drugs. And also, bioprinting itself can have so many controls. You can create so many different structures at your will. You can really do a lot of things from drug discovery and screening perspective. So I know that you did a lot of work in Alzheimer, Parkinson models, and as well as now tumor models. And most of these models are for drug screening purposes. Are there any intention of creating some kind of tissue, that can be implanted to repair people who are already got stroke or spinal cord got resected, that kind of thing?
Yeah, it's sort of interesting what goes to clinical trials. I think Lorenz Studer's group is taking dopaminergic neurons for Parkinson's. I think he's going to be doing transplantation either this year or next year. Who bought Geron? I can't remember. Asteris, is that right? Who does the spinal cord injury cell transplantation with embryonic stem cell derived oligodendrocytes. There was a couple of cell therapies for spinal cord injury which showed promise, but one of the big issues with that is cost. So I think in the Geron Asteris trial, I think all the patients, it was like something like $8 million a patient, just like really, really expensive. So even when you see some positive effects, like getting things like that adopted as standard of care is very hard. So from our perspective, it's sort of interesting because with the bioprinting, we were able to shift a bit. Previously, when I first started my lab, a lot of our work with the IPSCs was more towards cellular therapy. So it was always like, don't have any animal proteins, just being like really careful about a lot of the stuff to make sure everything was defined. We have been, though, playing with other things now as for drug screening, because now we aren't necessarily limited essentially you can add different molecules and things and animal-derived products to your bioinks to get better survival because we aren't going to actually be putting them in humans. So, but I think
go ahead. Sorry.
they just sort of inform each other, right? Like we looked at the protocols that make, purified populations of neurons, and obviously the work we do can also inform, how to differentiate and things like that. So sort of the two different sides of the coin. Also, I think just with the drug screening stuff with the patient HIPSCs, for example, because our collaborator, Dr. Nygaard, is a doctor, if we were to see a drug that would work in our model, he could potentially prescribe it back to his patients. And so and also I think just from a regulatory standpoint, it's a lot easier to get a drug approved than it is to get a cell therapy approved.
Yeah, that makes sense. And also the economics makes sense. It's probably a lot more cheaper to do drug screening using bioprinting than printing a piece of brain tissue or spinal cord for implant.
Well, there's like a whole group of ethicists, especially with the organoids, because you can recapitulate, and it's really cool. We've done it in our lab. We just haven't published it. Where you use the intrinsic power of your pluripotent stem cells, and you can grow these mini brains, and you put them on shakers and things like that, and they really do replicate the different structures found in the brain. But there's a group of ethicists that are concerned about like how large you should make them, and if you actually create these engineered brain tissues, are they thinking, and how would you know if your tissues were creating thought?
Yeah, I mean, I think the bioethics side is a whole brand new world that people never thought about stuff. Like even coming down to, how do you control the stem cell to make sure that the stem cells are going, are absolutely turning into the kind of cells that you want to induce them to. That is, I'm not sure how accurate we are at all times. So if we're going to do it in human body, there's going to be potential issues. You don't want to grow an arm out of, your chest or something like that.
All right. Well, the big concern for a long time was you'd get tumors. Like you'd get one of these cells be a tumor.
Exactly.
But yeah, the mini brains and the ethics is very interesting, and it's interesting to talk to my colleagues about it. It's like my favorite thing to do when we have drinks is to ask people like, do you think we're actually creating like thought in a dish? Because some people are just like, nope. Like everyone's oversold, overhyped these tissues. They aren't really recreating brain function. And then there have definitely been certain groups that have been calling like, you shouldn't grow tissues over a certain size and things like that. So, or
making a robot that looks like human. I mean, these discussions, I find them very fascinating and entertaining, and also necessary in humanity. Without them, we're probably not going to have computers, and we're not going to be here doing video conferencing. I think the human imagination has created a lot of positive things for us. So I don't mind I love talking about them as well. But definitely there are some scary futures that you could potentially, foresee, like my background here, Westworld.
Yeah, I'm always like, well, Westworld, that's not how you would print a horse. But anyway.
Yeah. Well, that's also coming from 1970s. The whole book was written decades ago. So, yeah.
Yeah, and I definitely know that there's some issues that arise, especially when you're doing some of the brain work where they're transplanting human brain cells into mice brains and things when we're doing some of the chimera stuff.
Or vice versa. Yeah, or the xenotransplant, right? That's like a huge discussion. Many decades ago, there was this, I hope I'm remembering this right, there was a kid who got a porcine heart because she was going to die if she doesn't have a heart. Her heart has defects. So I think she survived a couple more days, but that's generated a lot of discussions behind it. And definitely this is fascinating. I think I can talk a lot about it and love to hear about it too. But let's go back to science a little bit more. In your written interview, you're saying one of the challenges that you want to be able to predict the structures that you guys are going to print. And I think part of that maybe is because also we have lack of material science knowledge in a lot of these new materials that you guys are creating as well. Is that something?
Yeah, well, I think it's just because 3D printing, especially for the tissues, is one, new, two, it's so interdisciplinary that usually you're getting, like, for example, I work with Aspect Biosystems a lot. I know you're familiar with them.
Yeah.
Spun out of, Conrad's group that's electrical engineers, and so they took that perspective when making it. And so then you need to also bring in like some of your materials people, and then also just because the field is so new, there's not really a standard of how you quantify the different properties of your bioink. And so for us, we're like, okay, this is the speed, and then this is what it prints. And then there's just a lot because of the material properties and how the materials interact, and just even depending on the type of printing. It's really a trial error process. Is like, can we print this structure? Can we put these different cell types where we want? And then just a lot of validation. But the technology is definitely advancing quite rapidly. So.
Yeah, I mean, I know like for metal 3D printing and stuff like that, you have simulation software that can predict what the mechanical properties are. I wonder how soon can we have that in bioprinting?
Yeah, well, I know one of my colleagues, Matt Kinsella at McGill, like his group has done some stuff about like how you should characterize like the rheology and like the printing and like the printing speed and things like that. And I think just some of that stuff will get standardized. But yeah, a lot of it's, just because the field's so new. And a lot of the initial materials people use, like alginate's very easy to print, but stem cells don't really like to grow on it. So
That's too bad.
yeah, so, but I understand why a lot of people use alginate, but like, that's why some of our work has been really unique, is because we've actually been able to print and keep brain tissue alive and get functionality. But we do have people from the whole spectrum of engineering in my group. So.
But I know that you have a new material that cells like to grow on. You want to share a little bit?
Yeah, so yeah, our material is based off of fibrin, which is essentially one of the proteins found in blood. And we've spent a lot of time, my team has spent a lot of time optimizing it. And we've spun off a company called Axolotl Biosciences, which we hope to be selling Bioink by the end of the year. Obviously, Corona's put sort of a wrench in the plans for now, but I think we'll be able to figure some stuff out and get back to our original science soon.
Yeah, and I also look forward to you pitch at our Pitch 3D event during 3D Hills 2020, in addition to your presentations on your work itself. So I'm very much looking forward. I always like to see professors turn into entrepreneur, and I mean, you're in a very good position. So is this company run by your student as a CEO, or how? Tell a little bit more.
Yeah, so currently I'm the CEO, and then Laura de la Vega, who is my rockstar PhD student, she's CSO, and we have Lila Abelseth, who did a lot of the work helping to develop the formulation. She both works for me at the University of Victoria, and she also is a co-founder as well. I think we incorporated, although the corona's made me lose track of days, we incorporated in March, and we have a production manager, and we had a full-time business co-op in spring. In the summer, we're going to probably have 4 full-time co-ops as well, to sort of get things off the ground. The initial goal was to package and ship by summer, but that was without my lab shutting down. We have a good deal. So with the university, we can actually rent my research lab space to the company to do our production runs. And so, yeah, and Josh, who was our business co-op in spring, and he'll be continuing on in summer, he's made our website and got us up on social media and stuff. So yeah,
cool.
It's kind of a crazy time to be starting a company.
Yeah, but there are a lot of really good unicorn companies came out of the 2009 crisis, for example. So as long as you have, a really good business strategy and you have a really amazing product, it will survive. And
there's also some really interesting things coming out now about the effects of the coronavirus on the nervous system. So that's
I was going to ask you about that.
Yeah, so they think it can infect human neurons, and there's some evidence that it might be transmitted via the synapses, which is crazy but interesting.
That is crazy, yeah.
So we're hoping to put in some grants, and actually the company also we've submitted a pitch to the government of Canada on this, being able to use our neural tissue models to trace, to study the infection, because although we've been pretty lucky in British Columbia where we haven't hit the overwhelming stage yet, after this initial wave, you're going to be dealing with the people who've recovered, and what they've seen is there's been a lot of people who post-infection have epilepsy, and so there's clearly a neurological component that's going on.
Yeah, I've seen some MRI study after the coronavirus infection, and people cannot differentiate from the actual virus infection versus some kind of secondary effect from cytokine storm and everything else. So, and it's not like the first time virus infect brain. But I think this whole coronavirus situation finally is putting a spotlight on how virus affect human body, like not just the lungs, but every single human body organs.
Yeah. No, I think that there's been some, I think Joseph Pinniger's group out of UBC, he's got some different organoids showing how the virus affects these different organoids. So it's definitely infecting a lot of things, but obviously the thing most people focus on initially is the lungs, because that's where it's going in and infecting and spreading.
I think what the other interesting opportunity could be is that we can use some of the tissues that your lab is producing and maybe other labs are producing as a drug screening tool for the coronavirus drug development. I mean, if we want to speed this up, what other tools do we haven't used?
Yeah, for sure. And I think they just, I think GlaxoSmithKline just put out the preprint with making the pseudotyped virus. So that's, it infects but doesn't spread. It infects but doesn't replicate. So you can use it in a BSL-2 lab, because one of the issues is finding like BSL-3 space. Like at the University of Victoria, we don't currently have BSL-3 space for working with live corona. And so the fact that we now have, you can actually you have the pseudotype, which is a lower level as BSL-2. I think it'll open up a lot of studies for now as, the universities slowly get back to doing research again.
Yeah, so I mean, we definitely have a lot of opportunities here using bioprinting to advance science and pharmaceutical research. I'm going to wrap it up a little bit with a fun question.
Sure.
You said you are a fan of Kansas football team.
Yeah, the Kansas City Chiefs. I'm from Missouri.
Yeah, are you from Kansas?
I'm from Missouri. Kansas City is in Missouri.
Oh, okay. That makes sense.
Despite what Donald Trump says, the baseball team and football team are there. So yeah, I was very excited they won the Super Bowl.
Okay, so I admit I'm not a super sports fan, but congratulations. And that solves the mystery in my head is, you're in, British Columbia for so long, right? Almost how many years you've been there?
Years now, I know. And before I was in the Bay Area for my postdoc.
I know, I thought that you were in UC Berkeley.
Yeah.
So yeah, it sounds like you're all over the place, and congratulations again for all your achievement, and very much looking forward to your presentation in a month.
Yeah, well, it's good chatting with you.